Welcome to STEAMFest 2017. I have no doubt that this room is filled with our nation's next doctors, programmers, artists, teachers, maybe even a governor or a senator or two. As a former teacher, I know that students with science, technology, engineering, arts, and math backgrounds are in high demand. And that's why this opportunity is so important. You get to learn from some of the best and brightest in their fields, and you get to see what opportunities await you as you think about your next steps in life. I'd encourage you to take advantage of every opportunity. Public education is a foundation of our democracy. It opens up doors that wouldn't be possible otherwise. STEAM jobs are some of the fastest growing, good paying jobs in Montana. And that's why it is critical that we provide Montana students with the tools that they need to get an education in STEAM fields. As your senator, I'm working to make higher education more affordable by fighting for stronger Pell Grants to help students better afford the cost of tuition. And I'm supporting the Perkins Loan Program so more students can have access to the education that they need to launch successful careers. It's these kind of investments that will build a brighter future. I hope you get a lot out of this event, and please do stay in touch.
Hi guys, can you hear me? Can you see me? So I'm so excited to be here talking to you today. I am a neuroscientist and a graduate student at the University of Montana. Before we get too much into talking about me and what I do and where I'm from and how I got to how I am, to where I am, I wanted to do a quick activity with you guys. So it's called Put Yourself on the Line. So what we're going to do is imagine a line on the floor somewhere in your room. And I'm going to make a statement, and you're going to put yourself on that line depending on whether or not you agree or disagree with that statement. So everybody will have to get up out of their chairs. So here's the first statement. I'm having a great day so far. So if everybody can get up and imagine a line and put yourself on that line. If you are having a really, really great day, put yourself all the way to the left. And if you're not having such a great day, put yourself all the way to the right. All right, so once you've found your place on that line, I want you to turn to your neighbor and talk a little bit about why you put yourself in that place on the line. All right, so here's the next statement. The next statement that you're going to put yourself on the line, whether you really agree or you really don't agree, is, I know what I want to be when I grow up. So if you know exactly where you're headed after high school, put yourself all the way to the left. And if you don't know, if you aren't quite sure what you want to do, put yourself to the right. All right, so once you found your place on the line, I want to give you another second to turn to your neighbor and talk about why you picked that place on the line. Okay, you guys can go ahead and find your seats now. So the reason I asked that question about knowing what you want to be when you grow up is that I am still trying to figure out what I want to be when I grow up, and that's okay. So now I am currently researching neuroscience. So if you don't know what the word neuroscience means, I study brains. And I want to tell you a little bit about how I got to where I am. So I am from Montana. I have to move. Born in Darby, Montana, and I spent most of my life in Darby. A little bit of time I spent in Florida when I was growing up, but I graduated from high school in Darby. I went off to college at the University of Great Falls, and that wasn't necessarily my cup of tea, so I transferred to the University of Montana in Missoula. I got most of my college education, my undergraduate education, from the University of Montana, but I also spent some time in Ohio. But I graduated from UM with a degree in communication studies and minors in psychology and human and family development. So I got my degree and I didn't exactly know what I wanted to do. So I worked for a little while, and then I ended up back in school because I thought I wanted to be a doctor for a while. And I didn't have any of the science classes that you have to take to get into medical school in my undergraduate degree. While I was taking classes at U of M, I volunteered in a research lab. I wanted to mostly improve my medical school applications, but it turned out that I really enjoyed doing research. There are tons of opportunities to get involved in research at U of M at any level and to any degree that you want to. We just started an awesome new neuroscience program for undergraduate students as well, which is really cool. So when I first started working in the lab, I worked with brain cells grown in a petri dish like this, which you can kind of see. And then after that, I earned 2 different fellowships so that I could get paid to do research. So the first one was a summer fellowship where I was paid basically 40 hours a week to do research. And then I earned another fellowship to do research during the school year as well. And both of those opportunities were in a lab that studies fruit flies. So I got to do some really cool things working in that fruit fly lab. The first one here, this is one of the experiments that we would do. These are 2 male fruit flies, and they're fighting each other. So we were studying behavior in fruit flies. I also got to dissect the brains of fruit flies, which you can imagine are pretty small. We use very, very, very tiny tweezers and a microscope, and then you can take pictures of those brains using high-powered microscopes. And so that's what this image is here. That's a fruit fly brain in a high-powered microscope. So I liked doing research so much that I decided to go to graduate school at U of M and get a PhD in neuroscience. And this is what I am currently working on, the research that I'm currently working on. So I study electrical activity in the brain using frog eggs and cells grown in dishes. So this picture here is a picture of the frog eggs. That's a bunch of frog eggs in a dish. And the reason we use frog eggs is because most cells have the same basic structure and function, so we can use non-brain cells to learn about how proteins in the brain work. And they're easier to work with than brain tissue itself. This is another picture of an experiment that I do. So this right here is a single cell. I've stabbed it with this really sharp glass tube, and then in this tube over here are 2 different solutions. And when I apply the solution, this is the electrical response in the cell. So that's kind of what I do all day. I do these experiments, and I analyze the data, and I read about what's going on in the field. So I study a part of the brain and a protein that plays a really important role in learning and memory. As you can see from this picture here, that there are
the other way too.
Okay, there are a lot of parts of the brain that are involved in learning and memory. The part that I study is called the hippocampus, and it's kind of in the lower middle part of the brain. So what is learning and memory? Learning is how we acquire new information, and memory is the process that results in the storage of that learned information. So when we encounter information in the world, it goes into our short-term memory. If we don't do anything with it, we will forget it. From there, it can go into our long-term memory, or it can be quickly retrieved and be remembered immediately. So what I wanted to do with you today is test your short-term memory. So I have a couple of activities to do that, and the first one, what's going to happen is a series of letters is going to appear on the screen. After they disappear, you'll have a chance to write down what you remember. So don't write anything down while the letters are on the screen. Wait till they're gone before you write. Okay, thumbs up if everybody's ready. Okay, cool. I'm going to get out of the way. All right, and they're not going to be there for very long, so here they go. Okay, go ahead and write those down. All right, here comes the second set of numbers, letters. Okay, go ahead and write those down. Okay, next. Okay, go ahead. No peeking. And the next set. Okay, write those down. Oops. Sorry. Here's the next set. Okay, go ahead and write those down. All right, here's the last one. Okay, write those down. All right, so let's see how everybody did. Here are the answers. So I want you to real quickly just total up the numbers of each group that you remember, the total number in each group.
Total number of letters.
Okay, so now we're going to do another test to test your short-term memory in terms of objects. So for this one, on the next slide are images of a bunch of different objects. I'm going to give you 1 minute to memorize as many of the objects as you can. Don't write anything down while the objects are visible. And after that 1 minute, we'll see how many you can remember. Is everybody ready? Thumbs up. Okay. Few more seconds. Okay, now see how many of those objects you can remember. Just a couple more seconds to write those down. All right, pencils down. Let's see how everybody did. So here's the list here. Apple, flower, cat, bell, mug, cake, glasses, hand, football, truck, screen, sandwich, balloons, present, bird, magnifying glass, dog, pencil, the number 7, a tree, a green flag, a trumpet, earth, a bicycle, and a book. So if you look at how many that you remembered, research has shown that we can hold about 7 meaningful pieces of information in our short-term memory on average. So a meaningful piece of information can be a number, it can be a word, it can be an object, or any other chunk of information. And we can use this research about chunks of information to help us remember larger quantities of information. So let's test this idea of chunking. So I'm going to read you a set of numbers. They're not going to be on the screen. I'm just going to read them, and I want you to remember as many as possible. After I finish reading the numbers, write them down. Okay, here I go. 9, 1, 5, 11, 2, 4, 6, 15, 10, 3, 7, 13, 12, 8, 14. Go ahead. How many do you remember? Okay, now I'm going to read a second set of numbers, and I want you to remember them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Okay, go. How many of those do you remember? You don't have to write them down. You remember all of them. So both sets of numbers that I just read are exactly the same. It's just that the second set can be chunked into one bit of information that you remember because it's something that we encounter all the time. So you guys can use chunking to remember lots of information. For example, like if you're studying for a test, put like pieces of information together, and that will help you remember more in the long run. So studying is a form of practice or rehearsal of information that's required to convert short-term memory into long-term memory. Without practice or without studying, we often forget what we're trying to remember. So one really cool thing that research has also shown is that when we practice, when we study, when we form long-term memories, we actually change the shape of our brain and the structure of our brain cells. So these images here above me are brain scans, an MRI, of a study that looked at taxi drivers in London. And in this study, this was before Google Maps and smartphones and all of that, when taxi drivers had to just find their way around without technology. And so what they found in this study, these taxi drivers who use this part of their brain, the posterior hippocampus, all the time to find their way around, the size of their posterior hippocampus increased with the number of years that they were spent driving taxis. So they changed the shape of their brain by using that portion of their brain. We also change the shape and size of our individual brain cells. So these images here are individual brain cells, which are also called neurons. And if you look at those, especially those two arrows on the top, in this study where they were looking at learning and memory at the cellular level, those portions of this neuron where those arrows are pointing actually got bigger as the test went on. So there's a lot of really cool things that we learn about with neuroscience research, and this is just a few examples of those. And there's also a lot of things that you can do with a PhD in a science field. One thing that I've learned in graduate school is that I like to talk about science more than I like doing the experiments themselves. So I'm actually headed into a career in science communication. There are a lot of different options for careers in science communication. Some of the famous ones that you might know of, Bill Nye and Neil deGrasse Tyson are some science communicators. But there's also other opportunities to communicate science. This is Rosina Beerbaum, who served on the United States President's Council of Advisors of Science and Technology. So she informs the president about science and technology issues so that the policies that we make can be informed by science. And then there's also other opportunities. For example, Suzanne Shaw works in a nonprofit organization at the Union for Concerned Scientists, who also advocate for science. So that's all that I have to talk about today. And
Let's open it up for questions. How about that?
Great.
Let's start with Cut Bank. They have their snacks ready. They've been feeding their brains. So let's hear from Cut Bank. Cut Bank, do you have a question for Jenny or two?
Do you have a question? What are ways that we
So
can
where
improve?
did you do, after you left U of M, you said you went to another state. What state was that?
That was Ohio, and I went there
Ohio?
for a national student exchange, and then
Okay.
came back to Missoula. Yeah,
What did you do while you were there?
I just took classes. I was a student there as well.
Other questions, Cut Bank?
I don't know if we have any other questions right now.
Okay, thank you.
Thank you.
Thank you.
Let's go to Ronan. Ronan, do you have questions?
They were just figuring one out. Hold on.
Okay. No rush.
Okay. They were wondering what other tests that you have done using brain cells. Is it just with the electrical? Or?
So I mostly study the electrical activity, but in different ways. So a lot of what I do is testing the effect of different drugs or medications on the electrical activity. So I'm looking for drugs or compounds that can increase or decrease the electrical activity. So that's a lot of what I do.
Great. Anything else, Ronan?
Well, I think we have one more.
Yeah, that's good.
This type of research, you guys hoping maybe find
Okay, you're going to have to say it again because you're cutting out a little bit.
Come on up here to the front real quick. She's coming up so you can hear her better.
Okay, great. Thank
Cool.
you.
With all the new experiments with brain matter, are you guys hoping to maybe solve for certain brain diseases like Alzheimer's, or are you guys just?
Yeah, so there's two goals for the research that I do. The first one is to just learn about how the brain works, but the second one definitely is to learn what happens when the brain doesn't work properly, or when there's disorders like Alzheimer's or schizophrenia. The protein that I study is actually widely involved in kind of everything that the brain does, so figuring out drugs that can affect that receptor, that protein, could have implications for lots of different diseases, for sure.
Great. Thank you. Let's go on up to Conrad.
Say at least hello that we're thinking of a question.
We are thinking of a question currently.
Okay. Your short-term memory isn't what it needs to be to think of a question. Okay.
No questions?
No questions right now. Well, we'll bring Jenny back on, and she can answer questions at the end also. So let's thank her for her presentation, and we'll move on to our next presenter.
Thanks, guys.
She'll be here for a while, though. Next, we're going to hear from Inimmune, which is the company career aspect of this. So I'm going to let our presenter introduce himself. Come on in.
Thank you. Hi guys, my name's Dr. David Burkhart. I am the I am. It's kind of weird because I can't hear you, but you can hear me. But that's all right. So I'm the chief operating officer and a scientist from a new biotech company called Inimmune. I'm also a research professor at the University of Montana. I actually split time between the university and the company. And I'm here to talk with you guys today about
The other way. Come this way a little bit. Oh,
this
go.
way. Well, I was going to go
Okay.
off screen so I could read the whole title.
Gotcha.
So I'm here to talk with you today about what Inimmune is, what we do there, and what kind of opportunities that could present for students like yourselves as scientists or looking for a career in the sciences. So Inimmune is a biotech company focused on the discovery and development of new immunomodulatory therapeutics for treatment or prevention of allergy, autoimmunity, infectious disease, and cancer. So that's a lot of words. Basically, what it means is using the human immune system to cure itself with using molecules that alter the way the immune system works. And let's move on. That's the wrong way. There we go. Okay. So this is just kind of the slides that I'm going to present. The Inimmune story, who we are, our team, why would we do what we do, which is our mission. What do we do, which is immunotherapy, which is kind of what I described to you just a moment ago about the immune system, using the immune system to cure disease. Where we want to go, and that's clinical, which means advancing research drugs into clinical trials so that they can advance to the market and actually help patients. And how we get there, which are partnerships, both in academia and industry, to advance the research. So the Inimmune team is made up of a pretty large group. Right now we actually have 23 people. This photo was taken when we were at 18. But we basically have a large group of seasoned scientists that have worked together for over 10 years. We all came from a research site in Hamilton, Montana, where GlaxoSmithKline had a research facility that they shut down about a year ago. And so we lost our jobs, but we saw this as an opportunity to transfer some grants that we had to the University of Montana, and also start a new company and keep the expertise of that team together, and see what we could do in terms of using our experience to create new medicines in immunotherapeutics. And so our team is made up of a lot of different types of scientists. We have chemists who make synthetic adjuvant molecules, which are molecules that stimulate the immune system. We have immunologists that work on looking at how the immune system can use those molecules to cure disease. We also have molecular biologists involved in evaluating how those new molecules that the chemists make impact immune cells. And then we also have pharmaceutical scientists, like myself, that focus on drug delivery and how we deliver those drug molecules to different areas of the body that need those immunotherapeutics. And then lastly, we also have members of our team that are project management professionals, and these are basically people who are really good organized leaders who help manage these complex research projects. So as you can see, there's a number of different roles for scientists and non-scientists at the company. So why do we do what we do? What is our mission? Our vision is to create therapies for the betterment of humankind. Our company is trying to harness the human immune system, as I said before, to create the next generation immunotherapeutics for treatment of diseases. The immune system is incredibly powerful. If you think about what it does every day to protect you from diseases, probably every one of you sometime this year probably caught a nasty cold. You were sick and down for a few days, and then your body recovered fairly quickly. Your immune system protects you every day from a host of different pathogens. And dysfunction in your immune system can lead to or be involved in a number of different diseases and disorders. So some of those that we are focusing on are immunotherapy to treat allergy, which is sort of a hyper-responsive response from the immune system to foreign particles like pet dander or ragweed or timothy grass or pine tree pollen. We also are interested in looking at autoimmunity, which is dysregulation in the immune system's ability to recognize itself from foreign entities outside the body. And so you end up having an immune response against your own body, and that causes the disease. And then also we're interested in looking at how the immune system is involved in cancer, where we are retraining or reprogramming the immune system to recognize and eliminate cancer cells from the tumors. So one of the things that we do and how we do this is we actually try to harness molecules that are naturally expressed on the surface of bacterium or on bacteria on their cell walls. And these are called TLR ligands. And these molecules, our bodies have evolved receptors in our immune cells to recognize these molecules to know when your body's being invaded by a bacterium or a virus. And so if you look at this little guy up here, those little red dots are sort of the the little molecules that our body has evolved to recognize. And then in the gold cups below, you can see where those molecules are binding to receptors that our bodies have evolved to recognize those molecules and know to mount an immune response against that specific bacterium. So what we can do is we can take these small molecules, or these natural molecules that are on these bacterium cell walls, and we can modify their structure to isolate and harness the good properties of these molecules to use them as new medicines. And on this slide, you can see some of the structures of some of the molecules we're looking at. As I said before, we've been working for about 15 years on new synthetic derivatives of these TLR molecules. And when we change the molecular structure of these molecules, it alters their properties, both good and bad. So we can optimize their performance for a particular disease or disorder. And we utilize an approach called rational drug design to see how the structure of the molecules impacts the biological activity at the receptors. And a number of the different molecules that we've made have already been shown to be safe and effective in humans. MPL is a vaccine adjuvant that's produced in Hamilton, Montana, and is part of a number of different vaccines. And we're also working on other immunotherapeutics that are involved in cancer. And then just to show you, I'm going to stay on this side, just to show you kind of how these technologies go from being research molecules to medicine that you can actually get in the doctor's office. What you have to do is you have to carry these through from discovery and optimization into phase 1, phase 2, and phase 3 clinical trials. And that's where you take something that was just a research molecule and you're going to now try to make it into a medicine. And in the phase 1 clinical trial, you actually have to demonstrate that the molecule is safe, first and foremost, safe enough to be administered to humans and know that you're not going to have toxic side effects. And then in phase 2, you're looking at does this new drug actually have the immunological impact that you want? Does it actually help improve the disease? And that's basically done in phase 2 and phase 3. And if you can pass through those clinical trials successfully, then you can go to the FDA and get approval for your drug to go to market and actually be used as medicines in the doctor's office. So the primary disease areas that we're interested in: food allergy, allergic rhinitis, which is like seasonal allergy to pollen and things like that. Those are our top two indications that we've already done a lot of optimization and preclinical evaluation for, and our goal is to move those into a phase 1 clinical trial in the next year or two. And then also we want to look at, as I mentioned before, autoimmune disorders and cancer immunotherapeutics as well.
I wonder, go back to that slide for a minute. I'm going to ask the students this, because I think these are fairly common things. If you have a food allergy, if you're allergic to some kind of food, give us a thumbs up so we can just get an idea. Anybody out there allergic to a food? Great,
Yeah,
couple,
there's a few.
How about allergies like, is it like hay fever,
Sure,
and that
hay
kind
fever,
of thing?
or you're allergic to your cat, maybe.
Allergic to cats, hay fever, that kind of thing,
Those are pretty common.
right?
Yep.
So these are the drugs that might be in the market. How long does it take to bring them to market?
It typically takes anywhere from 5 to 7 years. So it's a pretty long process from discovery to get something all the way through to the doctor's office. But the interesting thing about this technology is that because we're retraining the immune system, it could actually potentially cure your allergy, whereas a lot of the drugs right now are just palliative. They just treat the symptoms. And so, you have to take a Claritin, or you have to take something like that, once a day or multiple times a week, this would actually be retraining your immune system to cure it of that allergy so that you wouldn't have to take anything for the rest of your life.
So in 10 years from now, if some of you who have food allergies or allergic reactions get to take a drug that cures it, you know the person who started it. So that would be great.
We hope so. We
Yeah.
hope so.
Yeah, go ahead.
So then the last thing I just want to focus on is how do we advance these potential drugs to the market and to the clinic. And that's industry and academic partnerships. And Inimmune has partnered with a number of different entities, including the University of Montana, UC San Diego, Canetta, and GlaxoSmithKline, to basically advance our candidate molecules into the clinical trials and get them approved as medicines.
Great.
And that's pretty much all I have. Thank you.
Okay, let's hang in there and let's go to each school for some questions. It's a lot of information, but I think it's important, students, for you to think about the pathway from the research that you do as a graduate student like Ginny to the industry. It's pretty exciting that it happens right here in Montana. So let's go ahead let's go to Conrad first. You. Conrad, do you have some questions about Inimmune?
What are you specifically working on right now?
So right now our focus is on allergy therapeutics and cancer therapeutics, and looking at how the immune system, how cells of the immune system can attack and eliminate tumors and cancer from the body. And the really cool thing about is using the immune system to do that is usually what kills a person from cancer is not the tumor itself. It's what's called metastases. It's the spreading of the tumor throughout the rest of the body. And because the immune system is everywhere in your body, it can recognize those cancer cells that have spread elsewhere in your body and eliminate them. And so it gives you a much better chance of surviving the cancer.
Wow. Other questions, Conrad?
So do they have any products?
Do you have any products in the market?
We do not. So as I mentioned, we're a very early company. We've only been in existence for 1 year because we used to work at GlaxoSmithKline. And so all of our technologies are still very early. We hope to get something there within 5 to 7 years.
Any questions?
Thank you.
Thank
Thank
you.
you. Let's go to Ronan. Ronan, do you have questions?
We do have one.
Can you give us a simplified version of how you can change the molecular type?
Sure, changing the molecular structure and that type of thing.
Yeah.
Yeah. So the simplified version is you get a chemistry degree. You go to college. You graduate from high school. You go to college. You get a chemistry degree. But seriously, that's what chemists do. Chemists are building these molecules from smaller molecules from the ground up. And so if we go back to one of the molecules where you can see a structure, the molecules you see on the right are pretty large. They have a lot of different atoms in them. And you, as a chemist, you build these from smaller pieces, adding them together, and then making modifications. And so we actually, chemists actually run reactions to make smaller molecules add together, and can change the shape and structure of the molecule that way. And the study of chemistry is studying all of those reactions and how they work so that you can control what happens, control the outcome, and get the molecule that you want to make.
Does that answer your question?
Yes,
Sorry
it
that-
did. Thank you.
Sorry, it's not more simple than that. It's a complicated process.
Great. Any other questions, Ronan?
Nothing else.
Okay. How about cut bank?
go?
Where do you get the funds for your research, like grants and stuff?
Yeah, that's a great question. So we basically we get funding from multiple sources. At the university, we're very focused on research grants. At Inimmune, companies can go after grants called SBIR grants. They're small business grants to basically develop new technology. So those types of grants are focused on discovery and screening, those early processes in drug development. And so we can write grants at the company to go get funds to do that. And then there are add-on grants for later stages in the process to try and advance things to clinical trials. Most of those grants come from the National Institute of Health, NIH.
Other questions, Cutbank?
Okay. So you're like talking about changing the molecular structure for like allergies and stuff like that. But could you prevent it like in people who have Alzheimer's? Like before the symptoms are like showing?
That's a good question. So preventing something before it shows is prophylaxis. That's prophylactic therapies. Therapeutic therapies are after the disease has already shown up. Alzheimer's disease is really tough with regards to immunotherapeutics and what we use for drugs because the blood-brain barrier makes it difficult for the immune system to traffic readily across from the rest of the body to the brain. And so it can be very challenging to try and cure something in the brain with the immune system. The immune system also is very poorly understood as to how it works inside the brain. We know there's some activity there, but we don't know exactly how.
Great question.
Yeah.
Anything else, Cutbank?
I personally have a lot of food allergies, and so do my people in my family. So are you working on things that just prevent those before I have like like allergic reaction, or
So food allergies,
or could it cure it?
Yeah, food allergies actually, we believe these technologies can actually cure it because your body is responding inappropriately to that food and thinking it's an invading pathogen. And what we're going to do is we're going to reprogram the way it responds to it so that it changes the magnitude of the response. So instead of giving a runny nose and runny eyes or congestion in your throat due to the histamines and all of that influence your body, it can actually retrain the immune system to know that food allergen is not as big of a threat. And that in essence would potentially cure it. And that can be done therapeutically. That can be done after you've already developed the food allergy.
Great question.
Thank you.
Thanks.
Thank you.
Let's bring Ginny in, and any further questions for either one of these folks? Just raise your hand. We've got two schools, Ronan and Cutbank. Raise your hand if you have any further questions for these two wonderful presenters. Okay, if not, thank you for attending STEAMfest. We know some of you will be back tomorrow, so let's give our presenters a hand. Thanks, guys.
Thank you.
Thank you.
Thank you.
Thank you.
Guys.